Hey everyone, my name is Sara Fumagalli and I'm a PhD student at the University of Milano in Italy. Today I'm going to tell you about the tool we recently designed, Madame, an easy-to-use tool for data and metadata retrieval in microbiome analysis. The study of human microbiome has been a hot topic for the last 15 years. However, the results obtained in individual studies cannot be compared with one another. Indeed, technical aspects, including experimental design and sequencing technologies, can bias the individual study results. To solve this problem, meta-analysis and secondary analysis are essential for the advancement of microbiome science by enabling to reach generalization to provide a more comprehensive picture that can be obtained from the individual studies and, of course, to overcome those technical biases. Clearly, to conduct this meta-analysis, data stored in the public repositories are crucial, such as those present in INA, NCBI, and DDBJ, that are established as part of the International Nucleotide Sequence Database collaboration. If we take a look at some numbers of the INA from the last year, we can see the huge amount of raw data stored and the great use by researchers who download this data every month. For this reason, in recent years, there are many tools that have been designed to download the data and/or metadata for public repositories, as you can see from this list, not complete. Tool specific to INA that allows the user to evaluate the suitability of metadata before downloading the data is still missing. Indeed, the lack of data harmonization and metadata standardization make challenging download the wanted data. Here I present you Madame, an open access and user-friendly tool that doesn't request the user to have specific programming or bioinformatics skills. It can be run through an interactive command line interface that will guide the user through the questions and examples. It can be easily downloaded from GitHub, where user can also find a detailed explanation of every step of the tool. And here's Madame's workflow. From a text query or a list of accession codes or a file containing these codes, the user can download the metadata in TXT format, where every row is a run. Madame uses ENA Browser Data API and ENA Portal API to read and download XML files, and Madame parses them into TXT files. The second module is the publication retrieval module. Here, the user can retrieve the publication associated with the projects that have been found in the previous modules. Madame searched for publication associated with projects in ENA, and if it doesn't find any record, it uses Articles RESTful API to search every accession code present in the merged metadata file in the European scene. The output of this module is a metadata file of the publication in TXT format containing several information, such as the link for publications, but also the DOI, the titles, the affiliations, but also text-mining terms. The third module is the record generation module. In this module, the user can view what he has downloaded in the previous modules in order to evaluate the suitability of findings before downloading the data. The outputs of these modules are an interactive report in HTML format containing plots and tables that are also downloaded as individual Excel tables and PNG images. Finally, the data retrieval module. Here the user can download the data in the three available formats available in ENA: FASTQ, XRA, and submitted using ENA browser tools. Let's take a look at the application of the tool. Starting from a fairly complex text query about the human skin microbiome, we search for projects. Madame created a folder for each project found and created inside an experiment metadata file, and optionally, if the user wants, a samples metadata file. And at the end, Madame merged all the experiments metadata file of all the projects into a single file, the merged experiment metadata file, that is the input of the next modules. So Madame found 36 projects with the metadata available and more than 8,000 samples. Then we searched for publication. Madame created a publication metadata file in the previously created folders and then merged all the publication metadata file into one. Madame found 33 publications for 25 projects. As a first step, we investigate if the results of the query were suitable and we generated a report. Here, an example of plot generated with the report module, a word cloud of the titles of the publications found. This is useful to give a quick first look and see if the query has downloaded relevant samples. For example, here we can see the words like oral, fecal, urine that make light a wake-up call. In fact, if we look at the bar chart of scientific names, we see there are many unrelated samples to our query that are indicated with those red arrows. Due to time constraints, I'll show you this latest plot, which is the worldwide distribution of the publication based on the affiliations of the authors, useful to understand the geographical origin of the samples. The report showed us a great heterogeneity in the samples that were found using the starting query, so we did not proceed with the download of the data. Anyway, I show you the fourth module, the data retrieval module. From the merge experimental metadata of all the projects, Madame uses in browser tools to download the data, and the data are downloaded into the previously created folders. In our case, all 36 projects have available data for a total size of more than 2 terabytes. So, to conclude, Madame results in a standalone tool able to meet a wide range of needs of the user without losing usability, thanks to its intuitive console. Madame offers instruments to further enrich information about data by searching the associated publications and visualizing it through the report generation, facilitating data examination, but also saving time and resources. Madame can easily provide the data and all the related information for downstream analysis, with the ultimate aim of contributing to microbiome research advances by facilitating multiple dataset integration. So, at this point, I'd like to introduce you the team and the colleagues who work with me. Unfortunately, the photos I found are all black and white, but believe me, they are very cheerful people. So I present you Giulia Soletta, Giulia Gossinetto, Manuel Striani, Prof. Maurizio Casiraghi, and Antonia Bruno. And of course, I thank you all for listening to me. And here you can find my contacts, and please do not hesitate to write to me. And if you are curious, I have also a poster. Bye and thank you.